I'm Eric Seemiller. I'm a vision scientist with the Operational-Based Vision Assessment Lab at Wright-Patterson Air Force Base in Ohio. Today I'm going to sort of extend some of the research that Dr. Ellie O'Keefe presented on Monday, specifically looking at some of the more interesting eye tracking data that we've taken. Specifically, we're going to talk about the near ocular motor response and how it changes perhaps to adapt to some depth distortions introduced by stereoscopic display designs. And hopefully by the end I will convince you of maybe a novel way of considering the ocular motor near response. Just real quick, these views are my own and do not necessarily reflect those of the Air Force, the DoD, or the US government. But I'm going to talk about, start off talking about what the near ocular motor response actually is. It's basically a suite of eye movements that occur when you are refixating your eyeballs on a target at far to a target at near, or just basically at two different depths. The first thing that happens is called binocular convergence. It's when the two eyes move in opposite directions. This is the international symbol for vergence right here. It's when the two eyes move in opposite directions of one another. And the goal of that is to align the two retinal images so that you have a nice, clear, single image on the retina. The second thing that happens is accommodation, and that's the change in the shape of the intraocular lens, and the goal of accommodation is to just bring the image into focus on the retina. It's to change the focal point of the eye. And the third thing that happens that I think a lot of people either forget or ignore or treat as a nuisance is pupil constriction. And that's basically, as it sounds, it's the pupil shrinks up. And the purpose of that is simply to increase the depth of focus of the eye, to perhaps relax some of the stress on the accommodative system to reduce the accommodative demand. Now for these three things, there is a direct neural linkage in the brain. So there are neurons that respond to blur that drive vergence. There are neurons that respond to disparity that drive accommodation. And there are neurons that respond to both that feed into the pupillary response. So there's really tight coupling in the primate brain for all three of these things, such that one cue can drive the other responses. And during natural viewing, this is fine. In the environment that primates evolved, these cues were rarely, if ever, in conflict with one another. Your blur signaled where your vergence needed to go, so that was all fine. However, in our unique situation, when we're designing stereoscopic displays, these cues can sometimes be in conflict with one another, or usually are in conflict with one another. The most famous example, the great unsolvable problem in stereoscopic display design, is vergence-accommodation mismatch. And you can see an example of that here. The disparity in the imagery is driving a vergence posture that is behind the screen, and the focal point is sort of stuck at the depth plane of the screen. So the accommodative response wants to go at one point, and the vergence response wants to go to a different point, and this can sort of reduce usability of that display in the extreme through that potentially uncomfortable conflict of those cues. Fortunately, there is that third arm of the near triad, the pupil, which can come to the rescue. To help increase the depth of focus and reduce the accommodative demand. So I'm showing you a sequence of retinal images that show the through focus going from -1 diopter to +1 diopter with two different pupil sizes. And hopefully this would be a 2200 Snellen acuity letter on your optometrist's eye chart. So this is a pretty big target, and you can see that if your pupil is smaller, much more of that through focus imagery is close to acceptable. It's not going to contribute to too much high spatial frequency defocus that will potentially drive an accommodative response. So basically, if the pupil comes in to help, the accommodative system doesn't need to work so hard. Despite these pretty well understood interactions of the near triad, we usually don't measure more than one at a time. We usually simply measure, vergence posture, or if you've got the right equipment, accommodative response, or if you focus strongly on human factors, you might ignore eye position altogether and just focus on the pupil. And so therefore we end up making assumptions about the ones that we aren't measuring. If we're only measuring vergence, we just assume the accommodative posture is where it's supposed to be. But I think hopefully I just showed you that that doesn't necessarily have to be true, especially with the third arm there. Fortunately for us, for most contemporary image-based eye trackers and photorefractors and even aberrometers, it's trivially easy to get 2 out of the 3. And the reason for this is because they all rely on an initial estimate of the pupil. So if you're measuring eye position, the first step is to find the pupil. And if you do that, you can measure it. So you have at least 2 out of the 3. And so the purpose of this paper is to sort of convince you that considering 2 out of 3 or hopefully all 3 if you can, you might be able to provide some insight into how humans are adapting to certain depth distortions and Q conflict that are presented in stereoscopic display design. So I'm going to talk about the experiment that Ellie talked about on Monday. I'm not going to go into too much details about what the task actually was, but it was basically telerobotic arm operation with a working distance in object space between 6 and 12 meters, so kind of far away. And your eyes were moving throughout this space. On any given trial, you could be anywhere between 6 and 12 meters away. So it was pretty robust in terms of where the eyes needed to go, and it was a precision task. So you needed to involve your eyeballs actively. We had 38 participants that were with all but one pre-presbyopic because, as we talked about Monday, promptly on your 45th birthday, your eyes stop working. And that's just noise that I didn't want to deal with. So we, generally speaking, stick to pre-presbyopic subjects, although one did sneak in there. And then we used the binocular eye tracker, the iLink 1000, which gets binocular posture or vergence at 1,000 hertz along with pupil size. And then eye position data were analyzed at the trial level and averaged within subject. Just to give you a sense of the distortions that we're dealing with here, so what I'm showing you is sort of the span of the object space where the target could be. In a perceptual image space here, so you can see most of the activities behind the screen. And there were 2 viewing distances. The near viewing distance was 96 centimeters. The far viewing distance was 170 centimeters, which led to almost a doubling of vergence-accommodative mismatch in the near viewing distance, which doesn't look like it in the picture, but diopters are weird, so it is true, believe me. So this is an example of the type of data that we would get here. The blue trace is the vergence posture, so you could see that this individual in this particular about 7-second trial made a kind of a sharp convergent eye movement and then diverged back to where they were before. I don't know what they were looking at or why they did this, I just know it happened. And then what you can see with the red trace is that a few milliseconds later, the pupil came along for the ride. We like to analyze these in velocity space. There's 2 reasons for that. Number 1, it helps to whiten the signal, which sort of makes downstream analysis a little easier. Number 2, we don't really trust the absolute calibration of our eye trackers, so it's kind of easier to think about this in sort of a relative space, which just makes velocity make more sense. We're just looking at the direction and velocity of the movement. So we convert to velocity space, just taking the first derivative of position, and you can see what that looks like right there. And then the way that we characterize the interaction between these 2 is through what's called a cross correlation. And we would just take the 2 velocity vectors and just kind of what amounts to sliding them across one another at different temporal offsets, and then computing the correlation of the two vectors at each of those temporal offsets. So we get a function of just the R value, just Pearson's R, as a function of temporal lag between the two. So for this particular trial, we have what you can see is a very strong correlation, almost close to perfect. It was like 0.97 that occurred at about 10 milliseconds after the change in vergence velocity. So I'm going to highlight a couple subjects for you just to show you what they did on close to 200 trials per session. So we analyzed the correlation at the trial level. So we have 200 samples per subject. So here are the peak correlations for this subject. In red is in the near condition, and blue is in the far condition. And what I hope you take from this particular subject is that at the near viewing distance, when accommodation vergence mismatch was highest, the pupil was extremely tightly dependent on what the vergence response was doing. Whereas in the far viewing distance, that coupling was somewhat relaxed. The pupil could do other things if it needed to. Those correlations are quite a bit lower. Wasn't true for all subjects. This is a different subject where there was much more overlap between the two, although I think it's slightly still higher correlations for the near, but it's somewhat negligible. For all trials and all subjects, there is a heavier tail on the right side there for the near viewing condition, suggesting to us that at that near viewing condition, again, the pupil response is much more tightly coupled with the vergence response. So what I'm showing you now is those correlations averaged at the individual level. So just the mean correlation for all the trials that a particular individual did. On the x-axis is each individual's correlation in the near viewing condition. On the y-axis is that response in the far viewing condition. And you can see they're pretty tightly correlated with one another. Not much error range in that least squares line, but the real take-home is most of the individuals fall below the unity line. And the unity line would be when their coupling was equal at both near and far. But you can see most of the individual subjects, not everybody, but most of them fall below that unity line, indicating a tighter coupling at near. If you know anything about the OBVA, we like to look for something you can measure in the clinic that would relate to this type of result. We looked at a couple things like stereo acuity, spatial vision aspects. We couldn't quite find anything that told a nice picture there, but we're still looking. We might try to look at certain elements of the accommodative response that we can measure in the clinic, but so far, nothing to say there, but ask us again next year. Just to show you that we thought about it, we also looked at the lag. Remember, each trial is characterized by the peak correlation and the temporal offset. Not really a clear story to tell here, sort of noisy distributions between 0 and 80 milliseconds, which, it sounds like a lot, but when you consider how slow and clunky both vergence and accommodation are, this is, it takes you almost a half a second for your vergence system to wake up and move to a new depth plane. So, 80 milliseconds relative to that half second is quite quick. But no difference between near and far is the take home here. So these results indicate a higher peak correlation between the vergence response and the pupil response at the near viewing distance, although not necessarily a tighter temporal linkage. What we think is happening here is this is a physiological adaptation to the Q conflict. The pupil may be doing its job in the visual system to help alleviate the greater accommodative demand that's caused by that VA conflict, forcing the or releasing the lens from having to work so hard and, interfering with the vergence response in the near triad. Whereas at the far viewing distance, the pupil has more leeway to do other pupil-y things like optimize ambient light level or, dare I say, come under control of the parasympathetic response if this is a stressful situation. Which brings me to my next point, is that when you are considering the pupil, there's a lot of folks in this field like to think of the pupil as some sort of window to the soul, but it is a part of the visual system. It's got a job to do, and that job is to optimize the retinal image quality. So, when you're thinking about an epinephrine flood from the locus coeruleus in the midbrain that's related to the parasympathetic response of the pupil, you also have to think about what the user's doing and how badly they need their visual system to behave and create a nice crisp image on the retina. So that's all I got. Any questions?